All right. Good afternoon, everybody. Welcome to the Technological Leadership Institute and to our webinar on cold supply chain challenges, biologics and vaccines. We're proud to be a part of IEM Innovation Week. Hope you're attending other events during the week as well. My name is Dan Bradian. I'm the Honeywell James J. Rainier Chair in Technology Management in TLI. I'm also the Director of Graduate Studies for our Master of Science in Medical Device Innovation. I am pleased to be joined by our three faculty that I'll introduce in a moment. But to get things started, I just wanted to note that few facts about cold chain management. The challenges of distributing the COVID-19 vaccine are the things that most directly have heightened public awareness about cold supply chain management. But the truth is these challenges aren't new, nor are they exclusive to mRNA vaccines. Cells, tissues, other small molecule therapeutics have their own limitations and storage requirements. Solutions to both near and long-term challenges in the field will require cross-disciplinary knowledge, leadership, innovation, and over the next hour, our panelists will address these challenges from their unique perspectives and roles in supply chain management. My objective as the moderator is to facilitate the dialogue between our panelists and you, so we invite each of you to prepare to ask questions. Our panelists will be presenting in order, and our first will be Allison Hubble. Allison is a professor in mechanical engineering and recently appointed director of the Technological Leadership Institute. We're pleased to have her with us. We're joined also by Tim Tripp. Tim is the director of cell therapies operations at Be the Match Biotherapies. And finally, Mac McKeon, who's a regulatory fellow at Boston Scientific Corporation. All three are members of our faculty, and we're pleased to have them with us today. Before we start, I'm going to remind our audience that if you want to ask a question, just raise your hand or use the QA feature to type in your question if you'd rather type it in than share it verbally. We'll be monitoring the chat box, the QA section, so that we're prepared when it's time to ask questions. So as I noted, here are our panelists, Allison, Tim, and Mac. And with that, I'm going to turn it over to our first presenter, Dr. Allison Hubble.
Thank you very much, Dan. I'm pleased to talk to you about cold chain issues. It's usually it's my biz, and so it's fun to be able to talk to people about my area of research. So we can go to the next slide. Okay. So as Dan alluded to, these are actually headlines out of the news. And so the fact that the COVID vaccine has been required to be stored at low temperatures has caused a bit of a buzz in the news and concern that this is something unusual, which, as Dan had already alluded to, it's not. So if we can go to the next slide. So let's start at 30,000 feet and say, why do we freeze things to preserve them? Because we could do all kinds of things. We could pickle them like we do food. We could salt them like we do food. We could do all kinds of things. Why do we freeze mRNA vaccines to preserve them? Well, let's go down from that 30,000 feet to the molecular level. Most biological systems are aqueous. They contain water. And one of the reasons why we freeze a biological sample is to immobilize the water. It goes from water in the liquid phase to an ice cube. So, and the reason why that's important is that water participates in a lot of degradation reactions. So by immobilizing the water, you prevent it from degrading the sample. So that's one molecular mechanism why we freeze things to preserve them. The second is that every system contains both molecules and degradative molecules, and those two interact to break down systems. And in our cells, et cetera, we have both DNA and DNases. And when we reduce temperature, those rates of reactions are slowed. So if we can go to the next slide. So those chemical reactions I'm talking about are a function of temperature. And if we can go back, Dan, they can be described by an Arrhenius equation. And don't worry, there won't be a quiz at the end of this talk. You won't be forced to derive the Arrhenius equation. But the reason why I put it in here is to show you it's very strongly a function of temperature. So for every degree that you drop, you get a lot of reduction in the activity. And hopefully there is a threshold activity below which, or threshold temperature below which no activity is observed. So if we can go to the next slide. So in our blood, we have roughly 15,000 proteins floating around. And each of those proteins has an activity with temperature. And clearly we haven't measured all of the activities associated with every protein in our blood. But one protein that has been studied is beta-glucosidase. And you can see in this lovely graph here that there is measurable activity for that enzyme down to very low temperatures, specifically down to minus 90 degrees Celsius. So if you were storing a sample that contains beta-glucosidase, we need to store it at temperatures at or below minus 90 degrees Celsius. So every molecule has a sensitivity to temperature and a temperature below which it is no longer active. So we're kind of setting the foundation here for understanding why a specific type of vaccine needs to be stored at a given temperature. That's because its properties and its rate of degradation and rate of reactions decreases with decreasing temperature. So can we go to the next slide? So here's our system of interest. These are the two vaccines that are out on the market right now. I know there's a little bit of kerfuffle with AstraZeneca and Johnson & Johnson right now, but these are the two mRNA vaccines that are being put in everybody's arms in Minnesota. I've been vaccinated, and my hope is that you are making plans to be vaccinated as well to help protect the herd in our state. There is a nano-encapsulated RNA, and the mRNA inside the cell may differ between the two vaccines based on the sequences that they're looking at targeting. But what you'll notice is that there are other things in the vaccine. There are some polymers and some structural components, including cholesterol, that act to control particle size and to reinforce the structure. And what you'll note is there's slight differences between the two vaccines in terms of the composition of that. And those differences in composition are why Pfizer is stored at a lower temperature than Moderna. That's one reason why the two are stored differently. So if we can go to the next slide. Another thing to keep in mind, it's not just these encapsulation and these structural components. What's inside that nanocapsule is also less stable. And that is as expected. Messenger RNA is less stable. It's far less stable than DNA. In particular, the ribose backbone is unstable. And that's the way it should be. Our RNA should be less stable than DNA. And so its stability reflects the nature of its biological function. And then, as said, there's a variety of different lipids, and they act in the nano-encapsulation, and there is variable temperature stability. Okay, so if we can go to the next slide. So when you're a manufacturer and you are working with or a biobank or any number of entities that work with biological samples and they're regulated, you are going to need to determine the shelf life of that item. And it's not a lot of fun, but basically you put it into a cold environment and you assay the stability with time and storage. And what you'll note is that in this, these are generic storage parameters, actually. These graphs mimic what you see for both DNA and RNA in -80 storage, just raw molecules. But this similar behavior is observed in a wide variety of other types of biological samples. So in this case, temperature A is lower than temperature B, and the shelf life for whatever you're storing is greater in temperature A than temperature B. So what that may mean, because we all have to compromise at some point, is that it may make sense for whatever reason, fit for purpose reason, that you need to store things at temperature B. And it could be that that's what has to happen for Pfizer to reach people in developing countries, for example. So that can be okay as long as you are able to live with the shelf life that you get from storing it at a higher temperature. And what you will note if you're looking through releases from the FDA is that the Pfizer vaccine has been recently approved for storage at higher temperatures, specifically storage at -20 degrees Celsius. But what that brings with it more than likely is a shorter shelf life. But the company understands what that shelf life is and are able to live with the shelf life that they have at that particular storage temperature. So there is my kind of molecular level to 30,000 foot level of storage temperatures and biological systems. So I'll turn things over to my next colleague, Tim.
Thank you very much, Allison. It's always tough to follow the better act. Allison is definitely a world leader in terms of cryopreservation in cells and biologics. So it's an honor to be serving on the panel with esteemed colleagues. And of course, Mac knows his regulatory stuff. We're going to learn a lot about that. I'm excited to hear him talk next. But it's a pleasure to be here. Thanks to Dan and the organizers for allowing me to come in and talk today. I've got my Johnson & Johnson vaccine in my arm, and it's been over about 13 days now, and I'm feeling fantastic. And even the peace of mind that it provides you by being vaccinated against this terrible pandemic has absolutely changed the world and really precipitated this talk today as we talk about cold chain and getting your product around the world in a pandemic that now requires freezing of product is amazing. And you know, doing it for the herd for sure. In order for us to get out of this, we all have to do it together. So I hope everyone's doing well. I hope you've got your vaccinations in. As Dan mentioned, I'm on the graduate faculty in the Master's of Science in Medical Device Innovation program. I really enjoy working with the students. So I hope I can't see everyone who's on the call, but I hope that we've got some MDI students and other TLI students in the audience today. And I'd be delighted to talk to you about your projects and about your work that you're going to be doing. And I'm always interested in serving on committees for your work. I really get a lot of joy out of that. So I just wanted to put that little plug in there. And Dan, I can see you, so like start waving if I go over time or anything like that. I'll try to pay attention to that. So yeah, so what I hope to bring today is a little bit of it was terrific to get the science behind this, and now what I hope to bring is a bit of the commercial component. Really, thinking about products and therapies that we have to move around the world and that we have to get to patients in need, and then understanding what the impacts of the cold chain has had on that and the changes that have happened. So Dan, if we could advance to the next slide, that'd be great. So as Dan mentioned, I represent a group called Be the Match Biotherapies. That is a function of the National Marrow Donor Program. It's a group right within the National Marrow Donor Program, which is a 30-year-old nonprofit organization that's facilitated traditional stem cell transplantation, bone marrow transplantation, really cellular therapy before it was even called cellular therapy. And really bone marrow transplant or stem cell transplant for those hematological indications is still the most widely used cellular therapy in the world right now. Thousands and thousands of transplants happen with stem cells from donors that go into patients with several indications. So the COVID situation has really put in a tremendous pressure on this entire sort of industry and everything that we're trying to do here. And it's been great. So a number of studies have come out. I'm just going to spend a few minutes on this one, which is from the Center for International Blood and Marrow Transplant Research, which is a function within the National Marrow Donor Program. You can see our cute little logos there with the green and blue. We have those kind of co-branding thing. CIBMTR is based out of here in Minneapolis. There's functions here in the National Marrow Donor headquarters, and then also functions at the Medical College of Wisconsin. And many of my colleagues were part of this work to really assess the impact of cryopreservation on donor grafts for outcomes in stem cell transplantation. So I'll take the next slide after this. So in order to try to keep it very quick, I'll just give a very high level overview of this. And this will kind of set the stage in terms of why we're starting to use cryopreservation, again, in traditional transplants. So these allogeneic grafts have been in the history of bone marrow transplantation and in this stem cell therapy infused fresh. It's a physician preference in the care of patients to really get those cells from a donor anywhere in the world to that patient anywhere else in the world within a very short period of time. We're talking 24 hours, 24 to maybe 36 hours. That's been the best practice. That's what physicians have really pushed for. That's what we're moving towards. Cryopreservation hadn't typically really been used in this step. So when was cryopreservation used? And this is important, of course, the inability to coordinate a graft collection. So a donor cell, somebody giving the donor cells, and a patient receiving them. These patients are incredibly sick. Their condition changes rapidly. They maybe have to wait a little while. And in those situations, you can't necessarily transplant that graft into those patients. So in that situation, cryopreservation has been utilized in the past. And the point of this of my slides is that in COVID, we had to really pivot and use it a lot more. So it's not well studied. There were small studies. Graft sources were often combined in the analysis. So sometimes you donate bone marrow stem cells. Sometimes you donate peripheral blood stem cells. So there wasn't a great body of research to say anything about the effectiveness and the engraftment of these stem cells. So in the next slide, Dan, I'll just quickly go over the study that was conducted just recently, 1,800 grafts or so, three cohorts in several different groups, really matching the case to control analysis. And then again, I know I'm going through this very quickly, but you can read the slide. But ultimately, the outcome of this study was that there was no significant impact in the cryopreservation of bone marrow grafts, but in terms of a few nuanced things like cryopreservation and platelets, there was some significant, we call it graft versus host disease. That's what graft GVHD is in the peripheral blood stem cell cohort. So we found that with utilization of cryopreservation, it changes your product. Your product performs differently in these different settings, depending upon sort of what the source of the cells and what you're actually going into. And then the same thing, inferior survival in the unrelated donor peripheral blood stem cell cohort. So, why did I take the time to talk about that? As in the new field, in the new world of cellular therapies, many of you I'm sure have heard of CAR T and other things. We really move forward with cryopreservation as our sort of default approach. And Dan, I'll take the next slide. And what it's really done is just, it has made a mess of the pandemic has made a mess of that supply chain in the COVID-19 situation. So in terms of how we do it, the courier availability. So typically it's a hand carried product, because again, these are personalized. You can think of this as personalized medicine. It's a set of cells for one patient. That requires an individual, somebody to actually courier to go and get those cells from where they are, take them to wherever they need to be. Hospital resources were very much taxed during the COVID situation. And then those patients in the bone marrow transplant ward could tend to be, sort of, I don't want to say ignored because we'd never do that, but they were deprioritized compared to the lines outside the door of respiratory issues. Huge issue there. International borders, was closures. We're still dealing with that to this day in the COVID situation. The airline flight, both cargo and passenger, flight reductions were hugely impactful to our, as we had to look at cryopreservation and getting these cells onto commercial carriers or passenger flights. And then transport requests went up as well. And then the unpredictable donor availability. I haven't gone much. I don't have enough time to really describe the model for biotherapies and our unrelated donor, but these are people stepping up, normal people like you and I, stepping up to donate their stem cells from their own body. And I don't really, in a pandemic, want to engage with the healthcare system and go donate my cells in a clinic because my COVID risk increases. So we'll just go to the next slide. And I probably am I over time already, Dan? How am I doing? Thumbs up, thumbs down? Go ahead.
No worries. Keep
Okay,
going.
good. So the critical thing in terms of us being able to continue to deliver these life-saving therapies throughout the pandemic, when we had to engage with cold chain and freezing the products in order to get them moved around, was really that redundancy and adaptability in our supply chain. And that's what provided the success that we needed in order to continue to deliver these life-saving therapies. If you look on the kind of the upper left there, cryopreservation and stem cell transplants, that's a great comparison from the same periods in the 2 years where the green is the cryopreserved. So you can see, in 2019, we were doing very few cryopreservation, only sort of as a last ditch result situation when the cells couldn't get to that donor, or the cells couldn't get to that patient from that donor in time. And then almost an inversion of that whole model in 2020 due to COVID-19. Through that, and because of our ability to utilize cryopreservation, we were able to continue to get these cells through. So you can see the 4,000 transportation legs completed, 2,292 transports completed. So many of these have to go from point to point to point, especially in the cell therapy, in the CAR T market. These cells are typically coming from a donor. They're going to a manufacturing facility. They're being manufactured. Then they're going back to a patient. So that's why you see transportation legs higher than transports completed. And through all that, really, there were just 2 missed delivery windows, and those had to do with overloaded cargo planes and the limited numbers of flights. So cryopreservation is absolutely critical in our supply chain. Our cold chain utilization so critical for us to continue to do that. So the redundancy and adaptability that we've built in, the broad network of partnerships that we have through relations, through customs and border protection, through multiple countries, so critical. If you have a life-saving therapy that is personalized, you need to have those. Those cells can sit and really expire in a customs cage at some airport we've been in Dubai to Shanghai to LA. Our managed logistics model is another one that from a commercial perspective, really having, I talked about that courier who's actually involved in going and getting that personalized medicine from that donor, from that patient, and moving it around the planet, essentially. That is a critical component for the success of our ability to get the cells to where they need to be. Our large collection network is also critical. So as you think about your supply chain and your organizations and how to mitigate against risks of pandemics and the utilization of cold chain, a diversity works, right? Diversity of locations is critical. So we have many, many locations throughout the world where we're able to collect cells to create these cellular therapies. And I've got a slide that I'll touch on just quick after coming up, that just how that actually totally played into this. I'll explain that later. And then really, as we've all done now, we're sitting here in a Zoom session. I feel like I'm talking to Mac Dan and Allison and Allie, and I know that there's many other on this call, but I can't see you all. The Zoom, in the commercial world, supplier quality audits are absolutely critical. I don't know if Mac will touch on this or not, but we've had to remotely onboard and audit these locations to provide us with compliant collections of material. Just an incredible burden. We had to scramble to figure that out. Typically, we would go to a site, collection center. We'd sit there for a day and a half, review documents, find their their quality systems, find compliance issues, write reports, and then remediation would happen, and they'd bring them on board. That's been moved to a completely remote world. We haven't gone on site for an on-site audit, and we haven't had anyone come on our facility for an on-site audit since the beginning of the pandemic, and that's been critical in your supply chain as you're thinking about your suppliers and how to audit them and keep up on them. Just is super important that COVID has done. And then, of course, in addition to that, we had the cryopreservation capabilities that we've had to add. So that was my point with that is, remote auditing cryopreservation capabilities and facilities has been a true challenge, but something that we absolutely had to do to deal with in order to move these therapies forward for patients. So, from the donor perspective, our large diverse donor registry pool with broad geography is helpful. And then again, the increased use of cryopreserved material really was the driver of all of this. It really gave us the ability to mitigate risk of that patient who's waiting for that cellular therapy to arrive to save their life. There's a great deal of prep work, of preparation that patient needs to do. I mean, you're doing a transplant, an organ transplant, except the organ is your immune system. You have to remove the old immune system and put in a new one. So these patients can't sit indefinitely without an immune system. I mean, that just does not work. They would so that our ability to then cryopreserve the material, maintain it for some time, get it to the location where the patient is, and then start the conditioning of that patient to receive that graft has been life-saving. It's literally been, cryopreservation has literally been life-saving for these patients. So my next slide, I just kind of talk about how we overcome that. So next slide, please. So all these things that I've sort of mentioned kind of get summarized in this cute little chart here. Stay-at-home orders. We had to, people had to stay at home, so that reduced all this unrelated and related donor travel. So we had a struggle getting our starting material, cellular starting material. Flight cancellations. We ended up with utilizing private planes connection, private aircraft. We actually had volunteer aircraft pilots ferrying these cells around the world to preserve cells to save the lives of these patients. The travel bans were just incredibly difficult. We absolutely, with the travel bans that came in place, we literally had to go to a nearly 100% cryopreservation approach because we had no guarantees that when those cells arrived at whatever destination they were going to be dealt with in a timely manner by those customs folks. Again, as we put them on commercial aircraft, removing that human element of the courier who can sit there and advocate for that therapy to get through. So we ended up obtaining CDC waivers and other travel waivers from other government agencies and partnering with hundreds of literally couriers and courier partner companies to get these cell therapies through. And then finally on this one is just the that international of the 40 or so cell therapies that we have moving around the world in any given day, half of them are crossing international borders. So it's really a challenge there. And as we started to try to understand how we could get cells to the US and get them moved around, we moved to what you see on here as a hub and spoke model. So we actually had many of our international shipments of cryopreserved cell therapies coming into Chicago. And at that point, we could put people in cars, they could drive to O'Hare, pick up the cellular therapy there, and then drive it around the country as that as the limited air travel was allowed. So that was just a great example of how we did it. So my next slide, I really talk about that donor starting material. So as we think about starting material for any process, really, how are you sourcing that starting material, and is cryopreservation going to need to be a component of what you do? One thing that we did to try to mitigate this was reduce ultimately the distance between that donor, someone who's providing donating their cells for the patient, have them geographically located near the patient that needs it to mitigate some of that risk. Cryopreservation is still incredibly useful in this situation, but the difference between our fiscal year 19 and fiscal year 20 was just really, it was a 30% reduction in terms of the distances that we had to travel. So I've got just kind of a quick summary slide and then one more after this, if Dan gives me the next slide. Thank you. So really, this pandemic supply chain picture, which we literally, cryopreservation solved. We looked at these barriers, our border restrictions, commercial airline options, just absolutely limited. Through the cryopreservation, that's kind of assumed in this entire slide, but this whole slide should actually just say cryopreservation. But the implementation of expanded travel options, these CDC waivers, our hub and spoke model, the corporate partnerships really allowed us to get 1,475 of these hub and spoke deliveries through April and into February of last year and this year. Those waivers were critical, working with the Center for Disease Control, getting those travel waivers for that cryopreserved product with someone carrying that was absolutely critical as well. And then those reduction in folks that were actually able to move those products around the world as well, and the restrictions that were placed on them. So we actually expanded what we have at the National Maradona Program. We call it our expanded, our employee courier program. Not only when you work at NMDP and Be the Match Biotherapies are you contributing to saving lives through cellular therapy, but you can actually grab if you ever see it, it's a blue box. It's called a Credo. Maybe somebody on an airplane carrying one of those. They'll have it very near to them all the time because in there is literally life-saving cells. And they're going to, one of those people was likely somebody who works for the National Maradona Program. So we had over 50 employees stand up and take on the risk, double mask, fly in these commercial flights to get these cell therapies through. So just incredible effort put in to move forward. So I'll just move on to my conclusion slide. Then, which is the next slide. So pandemic model absolutely forced our traditional transplant into a cryopreservation supply chain model. And all the science behind it was absolutely critical. The research that we've done to get to that point was just really needed. It's not perfect, so we need to continue the research on cryopreservation, especially those peripheral blood stem cells, to make sure that those work. And unfortunately, those are the key targets for like CAR T and other advanced cellular therapy starting materials. So the international border crossing increased the risk to the product and therefore to the patient as well. So all the things that we had done to cryopreserve and then allow us to have more time to get through those international border crossings was absolutely critical. And then finally, my last point is the commercial cell therapy developers must utilize cryopreservation to manage this supply chain risk. I mean, don't set up your facility in Shanghai if you're going to be collecting your cells in Chicago. I mean, the transport times and the certainty with which you can obtain either commercial or passenger air travel is a risk to your supply chain, and cryopreservation is going to have to be a component of that. So I'll stop there. I don't know if I went over. Dan never put the hook out, so I guess I'm good.
No, it's
the Mac now.
It's all valuable stuff. Thank you, Tim. So we'll now turn to Mac McKean.
Hello, everyone. My name's Mac McKean, and I guess I'll pivot a bit to this discussion and give another perspective on the medical device side. Whether I'm a third wheel or bonus round, I'm not sure, but appreciate Allison and Tim's presentation. Allison, that was fantastic. I'm very curious about just that whole vaccine thing and the structure. So I learned a lot, not that I'll remember any of it, but thank you for that, that lesson in biology, I suppose. So what I'd like to spend the next few minutes on is really considerations for medical devices in the design, manufacture, and distribution of the product, really across the total product life cycle. As you can imagine, if you're in the medical device business or even a recipient of a medical device, there's temperature restrictions, both high and low, for that apply to medical devices. So next slide. So really just to build on this total product life cycle, when you think about end to end, you've got really the design on the front end, then you've got to manufacture it, as Allison was talking about, and then the commercialization, right? Putting it into the wild, getting it to the patient, getting it into arms, so to speak. So really those considerations start for temperature and environmental conditions at the very design inception of your product, right? What are those environmental exposure limits for really the supply chain, but more importantly, the product itself, right? So your material selections, I mean, polymers are pretty tolerant. They don't have any water in them. I don't think they're going to move anywhere, frozen or overheated. But important considerations in devices are those products that include tissue substances or even combination products that will have a drug constituent, right? Those are all going to have upper and lower limits, right? It's almost like process control. What's your upper control limit, lower control limit? You just assign temperatures to that, right? It's reasonable to think that if you have a bioprosthesis valve that's soft tissue, that you can't freeze it, right? Or that there's a drug constituent that is temperature sensitive. I think that's grounded in the science and application of that product. So as you move out of that design phase into the manufacturing, make sure that your manufacturing process is also aware of and sensitive to the environmental extremes, both high and low, and what you expose your product and materials to as you manufacture it. An important part of manufacturing, of course, is the sterilization. Certainly, the vast majority of medical devices and products are provided sterile, of course. So they have to undergo sterilization methods. And there's many different methods that have very extreme temperatures, ethylene oxide, hydrogen peroxide vaporization, even steam, right? There's no cryo sterilization methods that I know of, but maybe we're working on it since we'd like to replace EO. So really, that all comes together in your design and process validation as your product moves through this total product life cycle. To validate that the materials and design and the bonding and the materials you've used will not degrade as a result of being exposed to high or low temperatures. So it all comes down to your packaging, design, materials, validation, shipping, and storage conditions. And I've got here just some clip art of temperature dots, right? A very easy visual. And of course, they're validated to the range of the temperature range for which your product has been specified and labeled, and a very easy indicator on the package for the user at point of use. Green, good to go. X, there was an excursion, don't use the product. Next slide. So really from a design control standpoint, and I thought I'd put a plug in too, I do teach MDI 5008, Quality Regulatory and Manufacturing Operations Management. And it's a very, my course is really built on what the process is that we all use in our industry, whether it's drugs or devices or biologics, is the stage gate model, right? So your user needs really come down to what are those environmental restrictions and exposures you're going to be into? What are you going to subject your product to? And then design and validate around that and go ahead and go through that model to the finished device. Next slide. Like everything we do, it's grounded in risk and risk assessment. If you're familiar with risk management tools, this is an ALART model, as low as reasonably acceptable. And it's a great visual, right? It's clear, green, yellow, red, what's unacceptable. So if you're doing your DFMEA, PFMEA for process risk, and then even the use FMEA, right, when it's out in the wild, you must assess the temperature excursion risk to your product and how the impact that'll have on the product, and then mitigate to it, right? Mitigate through your design, mitigate through your manufacturing, and then mitigate through your shipment and supply chain model. Next slide. ISO 1345 is very prescriptive about environmental considerations and talks about having specifications documented and procedures and processes to address and include packaging, shipping containers, specifying environmental criteria such as temperature and humidity. So again, these are all rooted in design, but find their way to the label. Next slide. This is a visual that I really like. I spent a lot of time on ethylene oxide sterilization. It's become a bit of a high profile issue of late. If you've been watching the news with the EPA, and they shut down some sites, and it resulted in some healthcare supply chain shortages, and we don't want any of that. But what I like about this slide is there's a lot of science. I should bring Allison into my class, talk about the science of sterilization, even at the molecular level, right? Because it's about killing the bugs, right? And EO is an effective agent to do that. But what I want to illustrate here is that while there's not a temperature range on the left axis here, you can see that in the 100 steps, there's a lot of temperature variation and relative humidity and injections and EO injections and vacuum draws and nitrogen washes and flushes and then aeration, right? To push off and draw off all the ethylene oxide fugitive emissions, right? Sounds like a prison movie, but we got to get rid of those fugitive emissions to make the product safe. So this is a consideration for when you do the design, right? So when you're picking your materials, make sure they'll survive your sterilization method. Next slide. So this is an example of labeling. Again, I won't go through all this, but as I mentioned, bioprosthesis and tissue valves, combination products like drug-eluting stents and drug-coated balloons. These materials have very tight temperature ranges in order to maintain performance per your validated design and the labeling. So as you can see here, there's some numbers references. They're pretty tight on the bioprosthesis because it's actually stored in a jar of glutaldehyde, otherwise called glute, because I don't think I can say it. And then also tight ranges for the drugs, because your validation work determines that this product will function to the label as indicated when preserved in these temperature ranges. Next slide. Next slide. There you go. So in summary, just to kind of pull that all together, again, the total product life cycle starts with design, selection of materials, make sure they can withstand and endure the temperature extremes of which your product will see, whether it's in the patient. Forgot to talk about the patient, but that's a pretty narrow range. I think maybe 90 to 105, the host then won't survive. But it's getting it to the patient for implantables. It's the key. Temperature indicators, as I indicated, must be calibrated to those ranges or they're not useful, obviously. And human factors are a very important part of this visually, right? So when it gets to the point of use and it's in the cath lab and they're ready to crack it open and put it in the patient, that green is good, let's go. Red is bad, there was a temperature excursion somewhere in the journey from your factory through distribution to the point of use. Again, that haul is validated through design, manufacturing, sterilization, shipping, and storage processes. And of course, your label, your device accordingly on those limits, and then putting controls in place with your couriers, right? So if you use Amazon, then you have a spec that it cannot exceed. If you're sending a valve up to Alaska in January, you can't freeze it or you can't use it, or likewise, August in Phoenix. So very intuitive to control that through the factory to distribution to the customer use itself. So that's my last slide.
Thank you, Mac. Excellent presentations all. We're gonna move into our Q&A session. And again, a reminder, if you haven't done so already, we're going to start with a few pre-selected questions, but we really want the audience to participate. So we encourage those of you who haven't yet to submit a question while we get started here. You can raise your hand if you want to verbally give the question, or you can type it into the Q&A session, and then Allie will make sure that I'm aware of it. But I have the moderator's prerogative, and that means I get to ask the first few questions. And I'm going to ask Allison a question, and that has to do with sort of the engineering and design, and ask, is it possible for you, wearing your research hat or Blue Q Bio perhaps, to actually design a system, either a cryopreservation agent or the system to manage it, to maintain a custom temperature or a specific temperature to meet the needs of a particular product?
Thanks for asking, Dan. This is actually a question that I've been asked more than once because, right now, as Tim so aptly described, these cells are, when they're cryopreserved, are shipped on liquid nitrogen. So wouldn't it, companies have come to me and said, wouldn't it be great if we could ship it on dry ice, or we could ship it with cold packs? And the biological system, whether it's a mRNA vaccine or a biological cell, will tell you what temperature at which it's stable. It's really determined by the biological properties of that material. Now, maybe we will get to the point where we can intervene and add a molecule that will neutralize something so that it can be stored at room temperature or some such thing, but we're not there yet. Certainly, it's something that's an active area of research.
Thank you. Thank you. And a question for Mac. You know, one of the things that we see during the pandemic is the rise in the use of emergency use authorizations, whether it's around diagnostic kits, ventilators, any number of products used in acute and even chronic care. And I'm wondering, you know, to some extent, those generally are have a loosening of requirements given the urgency. Does an emergency use authorization say anything specifically about temperature limits or restrictions on product use that would be relevant to this discussion?
Absolutely. I think that's a really important question, particularly with the pandemic and the hundreds. I think there are over 1,000 emergency use authorization clearances approvals at FDA. And I think Allison actually is really the best example I can think of, where the data that goes into the FDA for their emergency use authorization review, much as we've all kind of been part of in the news as they go through these vaccine reviews, right? Because it's headline news when they submit it to FDA and then everybody's staring at FDA, right? And a week later, they get through their DSMB and they make the determination. It took a little longer for some, but I think the important point here is all that data is in there, right? Now, FDA is not going to have the time, because it can take 6 months to a year to delve into deeply all the PMA data in a typical review, but this is expedited, right? And so what these companies do, whether it's a medical device or a vaccine, is they'll do accelerated aging, right? They'll do, certainly in the device world, you'll subject the product to elevated temperature and humidity to simulate. So in 3 months, you can simulate a year of shelf life, right? So that data is in there, but it's also early. And the point here, I think that Allison made clear, was that Pfizer was continuing to do that testing, environmental testing, the temperature testing, in parallel to not only the EAU, but the implementation of this first round of vaccines. And now they've raised the temperature, right? Because they have more data. So I think that's the interesting thing about the EAU process, is it is iterative. FDA is acting on the data set that they have, and they'll label accordingly. So that's why when the Pfizer came out, it had that uber cold restriction, because they didn't have the data to say they can go to 20C, minus 20C. So I think we're seeing that in action. And it's a continuous review model. And I really applaud FDA for the great work that they've done across the COVID and EAU and other tools to really do everything we can to have access to therapies.
Excellent, thank you. Question for Tim, and Tim, this is actually a question triggered in part by your excellent presentation, as you described the need to really just fundamentally change your supply chain model. I'm wondering about the impact on expense. You're the director of operations, so I suspect you're sensitive to such things. But you've shared the data around the impact in terms of time, et cetera. I'm just wondering, in general terms, how it's impacted your expensive operations and how you manage that.
Yeah, absolutely. So you can see from the slides that I had, with fresh product being the typical approach, that is tick panels and accredo. I mean, these are literally those blue things that you'd use for putting in the cooler if you're going to the beach. It really is about that primitive in terms of the fresh product shipping. When you start to talk about utilizing dry shippers, it becomes a very different story in terms of the costs. So we have seen a tremendous cost increase in terms of getting our products to the patients in need, and that comes through utilizing the like the SAVSUs and the Cryoport dry shippers that are out there for these kinds of applications. And not only are you then you're carrying the burden, the cost of that capital, right? This is a instrument at that point. I mean, it's capital, it's hitting your capital budgets as well, but you complicated your supply chain because you have to get that back and you have to reuse it. So not only have you sent something out into the world to be utilized by who knows who, but if they have damaged it or used it improperly in any other way, the reusability of that becomes a big risk as well. So we've had to add in central locations to receive these dry shippers back and then send them out to these other sites for cellular therapy collection. So that has, not only is just the cost of getting those and using them been an additional burden, but the additional complexities of getting those back and utilizing them again has also been something that we haven't experienced, and it's definitely added.
Well, I've got a question.
Dan,
for,
can I?
Well, Allison.
Yeah.
Oh,
Dan,
go
can
ahead. Go ahead, Allison.
can I piggyback
Yes,
a
absolutely.
little bit on
Yeah.
what Tim had started? And this kind of addresses Jeffrey Tate's question in the Q&A, that any kind of temperature controlled shipping, you're right, extremely expensive, heavy. The other thing that's complex is sometimes, and this happens in biobanking, in which you may have one type of specimen that needs to be stored at one temperature and another specimen at a different temperature. And those kind of shipping arrangements are also very expensive. So this is a need. So this is a need for new technology and to reduce weight and cost. I would note, however, since there are some potential students in the audience, that we have Minnesota-based companies who are actively involved in research and product development, in particular CHART, which used to be Minnesota Valley Engineering down in either in Shakopee or southern of that in New Ulm. They're very actively involved in research in that area, and they're also looking for really good engineers who can address those design needs.
Yeah,
Allison, thank you.
adding that, Allison, and really some of these dry shippers, they're the size of a refrigerator, and many times in cryopreservation, certainly in experiences that I've had, if you're moving your lab or your research function, sometimes you just duct tape your -80 freezer shut, and you ship your entire -80 freezer across the country, and that's also a kind of a workaround to this complexity of these devices. I guess calling them devices as these dry shippers and things like that. So it's really ripe for experimentation, for research, and for product development for sure. Because again, dry shippers, they lose their charge over time, and if after 36, 48 hours, 72 hours, your product that you've spent all this time and energy and money collecting and getting and putting into this very expensive, very large dry shipper, if it sits on the dock at the airport for longer than that, your product is ruined in the personalized medicine world, where one patient for one product, that's devastating. That's devastating.
I think another important point there is that duct tape you use is 3M duct tape, just to put in that local.
We've got a question from Winnie Medina, who it turns out is a member of the incoming MDI class of 2021-2022, and she asks, as promising and as effective as the new mRNA vaccine technologies have been, they're currently approved under emergency authorization. What are some challenges you anticipate in a standard approval process? and having to do with user needs, overall business model. And so I don't know if Mac, if you want to take a shot at this, it's sort of a follow-on to your own comment, and then I'll open it up to Allison and Tim as well.
Sorry, what was that? What was the question again, Dan?
Well, basically that you know the new mRNA vaccines, at least presently, are under an emergency use authorization, and you note that the data is being collected, and yet, you know, if they were to seek complete standard approval right now, one would anticipate they wouldn't have everything they need. So, what are the likely challenges that you see for them?
Well, I think it's a data-driven, well, I know it's a data-driven process. And I liken this to, it kind of came to me as I had answered that, or made that comment earlier, this is kind of an expansion of indications, right? So what this does through the EAU and feeding, you know, not piecemeal in a bad way, but giving data sets in parallel to FDA allows you to expand the use population potentially. But in the case of this example, in terms of temperature, just think and really tapping into Tim and Allison's big picture, whether it's 30,000 feet or the airplane image, is now Pfizer, their distribution chain is much easier and broader, right? Because they go from what was it, minus 40 C to minus 20 C. So the logistics around that, I think, are crucial, right? I mean, you could probably do an analysis and really understand how much that is further facilitating access, even to rural areas, right? I'm not trying to play CDC here, but that's just such a great example about this whole talk, what just recently happened with Pfizer. And, you know, maybe they continue to do this work and get that to zero, or, you know, and whatever, but they're not, you know, I'm not the pharmacologist here. They're not tinkering with the RNA, right? I mean, they have formula. It is the formula. You don't change that, right? But you understand it enough to go get this other data, whether accelerated or real time. And I think that's an important point, real time data, right? Maybe some of what they presented through the EAU update was, hey, here's real time data that while we exposed some of these to these temperatures, it was still potent and effective. So.
Excellent. Allison or Tim, briefly, anything to add on that?
So just one other thing to kind of undergird our thoughts here. mRNA vaccines have been standard of care in the veterinary field for well over a decade. So this is well-tested technology. It has entered into the human phase. And my hope would be, in terms of exactly what Max said, that we're going to find this vaccine to be effective over all ranges of age, so that everyone in the, all children, adults can be vaccinated with the same vaccine. That's what I hope happens in the future. That's what I hope they're waiting for to get the standard approval, is to have as broad an umbrella as possible for this technology.
Okay, very good.
Yes, really interesting stuff. I mean, certainly a pediatric approval will be a challenge out there, I think, for the industry to deal with, in terms of moving the mRNA vaccines into younger populations. And then the long-term follow-up is going to also continue to be an issue. Look at how great this is, though, to have, you know, ultimately a phase 3 clinical trial with numbers like this is fantastic. So we'll absolutely be able to have a great set of data, which may even become more challenging, be really a kind of a challenge in the future, is actually analyzing all that data. We look at what happened with Johnson & Johnson in this very, very rare situation. I mean, 0.000, I think 4 zeros, and then an 8.
It's one in a million. So that's pretty good.
Yeah, I think I'm buying lottery ticket. You know, my daughter.
It's 10 to the minus 6, right? That's what
10
we
to the
do in
minus 6.
sterilization. That's pretty good.
Yeah, it really is, you know, but here it is. We are a risk averse society in the US, and we want to be extra cautious about these things. And, you know, first,
I think what's intuitive and applicable to all this is all this data and technology moves on to next gen, right, Allison, as they do the next version of this, right? And the protein analysis, all this temperature data and these extremes and that real world evidence carries forward that knowledge for which to build on to better serve a broader population.
And with that, we're going to have to wrap up here because we're almost at time. So I want to thank our panelists again for wonderful presentations and thoughtful discussion afterwards. I want to thank staff, Allie, and our marketing department and others, obviously, for putting this together. It's been my pleasure to be the moderator. I want to invite folks in the audience to visit the TLI website and to follow us on Twitter, Facebook, LinkedIn, and YouTube as we post videos of this and other of our webinars. And for those of you in the audience who may be interested in our program, particularly our MS in Medical Device Innovation, just a few notes. It's designed for working professionals. It's a full-time program that meets in the evenings and occasional Saturdays. We help you develop the skills in innovation, biodesign, medical product development, and leadership that will help differentiate you in the marketplace. It's a cohort-based model with enrollment limited to approximately 30 students once a year. Course schedule Mondays and Wednesday evenings with one Saturday per month. Our next cohort begins in the fall of 2021. And so we're still actively recruiting. And we've got new program options. We've got our traditional 16-month or 4-semester option, but we have introduced an extended option for folks that want to take a little more time, an extra semester to complete their study and particularly their capstone. And so we encourage interested parties to join us at our next info session, which will be Thursday, April 22nd from 5 to 6 online, and you can register at TLI. So again, thank you, Allison, and welcome to TLI. Thank you, Tim. Thank you, Mac. Really appreciate your efforts today and appreciate the fact that you take time to serve on our faculty. With that, I'm going to turn it over to Allie and wish all of our attendees a pleasant afternoon.